The fundamental question that we're trying to deal with has to do with the fact that the sediments of the oceans are teeming with life. And so the organisms, whether they're bacteria or very, very small eukaryotic organisms, they're tremendously diverse. And we know that they're doing very important functions in the ecosystem. So they're very important to the transport of organisms around in the sediment, to breaking up the sediment, to mixing it, and fundamental processes of metabolism. So converting, for example, hydrocarbons into other substances. And they're an essential part of the food chain. Our particular interest of our group, our team, is on the eukaryotic fraction of those, so the animals primarily that live there. And so those ocean sediments are tremendously diverse. The problem that we face is that as they are the ocean sediments, they're vastly understudied. And so really almost anything you find there has never been seen before. It's never, people can put general names on them by body plan, but they really have no idea what they are. And really don't have a specific idea for any particular organism what they do. They know what the group does, but they don't really know by organism. And those, of course, the issue that we're faced with in the context of this community that we work in is trying to understand the changes to those communities that come after a perturbation, like an oil spill. So how you have to understand what was there, what they were doing, and then what was lost or gained, and what was lost and gained and what that community is doing after that. Our approach and the approach to solving this problem of who's there and maybe what they're doing has been transformed over the last few years by the new genomic technologies, the ability to sequence DNA accurately, rapidly, and for almost nothing. Right? Of course, driven by personal genomics and all of the other areas of importance in human health. And so we're applying those tools, those technological tools, to this question. And so what we would like to, this is a theme for project, whether that is meaningful or not. And so the project is aimed at developing the tools to be able to assess in a rapid, accurate way how those communities are changing over time. And so really it's a development of that approach. And so we're going to apply the genomic technologies to that, and we're going to apply the bioinformatics that are necessary to analyze that. The critical part of this and the thing that's holding us back isn't the ability to sequence DNA or even analyze it right now. It's that we need some references. We need to have reference examples, at least from the breadth of the phyla that exists there. And so the first phase of the project is to with my colleagues, the team, is to go and sample from the Gulf of Mexico specifically, and sample hundreds of different species and develop genome sequences for those species so that we have something to compare to and look at that. And so that's the way we're going to approach that. And then ultimately, we would hope to be able to apply that technology to sediment cores themselves, to sediment samples before and after spills. I have two colleagues on this grant. One is Holly Bick, who is an expert in these groups of organisms that live in the sediment, and on the application of and development of bioinformatics tools. So Holly is a key part of this program. The other person that is very important and his position in the Gulf of Mexico and his research institute, which is the HART Research Institute for Gulf of Mexico Studies, this is Paul Montagna, and his work is fundamental to this in terms of the actual collection of these organisms for developing this reference database. We're also conducting, almost all of the activities are taking place at the HART in Corpus Christi in terms of things like not only the biological specimen collection, but the workforce development that's necessary to make these tools a reality are all going to take place down at the HART. And so we think it's a great opportunity to do that.